ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. ಕೆಂಚಾಗಿ ನನ್ನ ಛಾಯಾ ನಿಮ್ಮೆಲ್ಲರೂ ವೆಲ್ಕಮ್ ಮಾಡ್ತಾ ಇದ್ದೀನಿ. ನಾನು ಕಾಲೇಜಲ್ಲಿ ಕೆಮಿಸ್ಟ್ರಿ ಪ್ರೊಫೆಸರಾಗಿ ವರ್ಕ್ ಮಾಡ್ತಾ ಇದ್ದೀನಿ. ನಾನು ಇಂಟೆನ್ಷನ್ ಈ ಚಾನಲ್ ಸ್ಟಾರ್ಟ್ ಮಾಡೋದಕ್ಕೆ ಅಂದರೆ ಸಾಧ್ಯ ಆದಷ್ಟು ಕೆಮಿಸ್ಟ್ರಿ ರಿಲೇಟೆಡ್ ಟಾಪಿಕ್ನ ಸಿಂಪಲಾಗಿ ಕನ್ನಡದಲ್ಲಿ ಹೇಳುವಂಥ ಪ್ರಯತ್ನ ಮಾಡ್ತಾ ಇದ್ದೀನಿ. ಕೆಮಿಸ್ಟ್ರಿ ಇಷ್ಟ ಇಲ್ಲದರೂ ಕೆಮಿಸ್ಟ್ರಿ ಕಡೆಗೆ ಸೆಳೆಯುವಂಥ ಪ್ರಯತ್ನ ನಾನು ನನ್ನ ಚಾನಲಲ್ಲಿ ಮಾಡ್ತಾ ಇದ್ದೀನಿ. ಸದ್ಯಕ್ಕೆ ನೀವು NEP ಏನಾದರೂ ತೊಗೊಂಡಿದ್ರೆ, ಓಪನ್ ಎಲೆಕ್ಟಿವ್ ಅಂತ ಒಂದು ಸಬ್ಜೆಕ್ಟ್ ತೊಗೊಂಡಿದ್ರೆ, ಪರ್ಟಿಕ್ಯುಲರ್ಲಿ ನಾನ್ ಸೈನ್ಸ್ ಸ್ಟೂಡೆಂಟು ಓಪನ್ ಎಲೆಕ್ಟಿವ್ ಕೆಮಿಸ್ಟ್ರಿ ತೊಗೊಂಡಿದ್ರೆ ಸೆಕೆಂಡ್ ಸೆಮಿಸ್ಟ್ರಲ್ಲಿ ನೀವು ಮಾಲಿಕ್ಯೂಲ್ಸ್ ಆಫ್ ಲೈಫ್ ಅನ್ನೋದನ್ನ ಬಗ್ಗೆ ಹೇಳ್ತೀರಾ. ಸೊ ಆ ಸೀರೀಸಲ್ಲಿ ನಾನು ಕಾನ್ಸೆಪ್ಟ್ ಆಫ್ ಎನರ್ಜಿ ಇನ್ ಬಯೋಸಿಸ್ಟಮ್ಸಲ್ಲಿ ಇರುವಂಥ ಚಾಪ್ಟರ್ನ ಈಗ ಅಪ್ಲೋಡ್ ಮಾಡ್ತಾ ಇದ್ದೀನಿ. ಆಲ್ರೆಡಿ 11 ವಿಡಿಯೋ ಅಪ್ಲೋಡ್ ಮಾಡಿದ್ದೀನಿ. ಇದು 12ನೇ ವಿಡಿಯೋ ನಾನು ಅಪ್ಲೋಡ್ ಮಾಡ್ತಾ ಇರೋವಂಥದ್ದು. ಇವತ್ತಿನ ವಿಡಿಯೋದಲ್ಲಿ ನಾವು ಲಿಪಿಡ್ ಮೆಟಬಾಲಿಸಮ್ ಬಗ್ಗೆ ವಿಡಿಯೋನ ಮಾಡೋಣ ಅಂತ ಅಂದ್ಕೊಳ್ತಾ ಇದ್ದೀನಿ. ಸೊ ನಿಮ್ಗೆ ಆಲ್ರೆಡಿ ಗೊತ್ತು, ಲಿಪಿಡ್ಸ್ ಅಂದರೆ ಏನು, ಟ್ರೈಗ್ಲಿಸರಾಯ್ಡ್ಸ್ ಅಂತ ಕರಿತೀವಿ ಅಥವಾ ಆಯಿಲ್ಸ್ ಅಂಡ್ ಫ್ಯಾಟ್ಸ್ ಅಂತ ಕರಿತೀವಿ. ಸೊ ಅದರದ್ದು ಹೇಗಾಗುತ್ತೆ ಮೆಟಬಾಲಿಸಮ್. ಹೇಗೆ ಮೆಟಬಾಲಿಸಮ್ ಅಂದರೆ ಎರಡು ಪ್ರೋಸೆಸ್ ಇರುತ್ತೆ. ಒಂದು ಅನಬಾಲಿಸಮ್ ಅಂತ ಹೇಳ್ತೀವಿ, ಇನ್ನೊಂದಕ್ಕೆ ಕೆಟಬಾಲಿಸಮ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಲಾರ್ಜ್ ಇದ್ದಿದ್ದು ಸ್ಮಾಲಾಗಿ ಕನ್ವರ್ಟ್ ಆಗುತ್ತೆ, ಮತ್ತು ಸ್ಮಾಲ್ ಇದ್ದಿದ್ದು ಮತ್ತು ಯೂಸ್ಫುಲ್ ಸಿಂಥಸಿಸ್ ಆಗಿ ಕನ್ವರ್ಟ್ ಆಗುತ್ತೆ. ಈ ಎರಡೂ ಪ್ರೋಸೆಸ್ಗೆ ನಾವೇನಂತ ಕರಿತೀವಿ, ಮೆಟಬಾಲಿಸಮ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಅದ್ರ ಬಗ್ಗೆ ಇವತ್ತು ಹೇಳೋಣ ಅಂತ ಅಂದ್ಕೊಳ್ತಾ ಇದ್ದೀನಿ. ಎಗೇನ್ ವೆಲ್ಕಮ್ ಟು ಕೆಂಚಾಯ. ನನ್ನ ಚಾನಲ್ನ ಸಬ್ಸ್ಕ್ರೈಬ್ ಮಾಡಿ, ಶೇರ್ ಮಾಡಿ. ನೀವು ನನ್ನ ಚಾನಲ್ಗೆ ಹೋಗಿ ಪ್ಲೇ ಲಿಸ್ಟ್ ಹೋಗಿ ಓಪನ್ ಎಲೆಕ್ಟಿವ್ ಸೆಕೆಂಡ್ ಸೆಮ್ NEP ಅಂತ ಕೊಟ್ಟರೆ ಇದುವರೆಗೂ ನಾನು ಅಪ್ಲೋಡ್ ಮಾಡಿರೋ ಎಲ್ಲೋ ವೀಡಿಯೋಗಳು ನಿಮಗೆ ಸಿಗ್ತಾ ಹೋಗುತ್ತೆ. ಚಾಪ್ಟರ್ ವೈಸ್ ವೀಡಿಯೋಗಳು ನಿಮಗೆ ಸಿಗ್ತಾ ಹೋಗುತ್ತೆ. ಹಾಗಾಗಿ ಎಕ್ಸಾಮ್ಗೆ ನೀವು ಪ್ರಿಪೇರ್ ಮಾಡ್ಕೊಳ್ಳೋಕ್ಕೆ ಈಸಿ ಆಗುತ್ತೆ. Lipids are available to the body from the three sources. They can be ingested in the diet, stored in adipose tissue of the body, or synthesized in the liver. Fats generally are triglycerides within the body are ingested as a food, are synthesized by the adipocytes or hepatocytes from the carbohydrate precursors. Lipid metabolism entitles the oxidation of fatty acid to either generate energy or synthesize the new lipid from the smaller constituent molecule. Lipid metabolism is associated with the carbohydrate metabolism, as product of glucose can be converted into lipids. Lipid metabolism begins in the intestine, where indigested diglycerides are broken down into free fatty acid and a monoglyceride molecule by pancreatic lipase enzymes that break down the fats after they are emulsified by the bile salts. So triglyceride, this triglyceride, when they undergo metabolism, it get converted into a monoglyceraldehyde, and also pancreatic juice and also bile juice, it going to emulsify fatty acids since they are insoluble in water. Once the bile salt is emulsified, the triglyceride, the pancreatic lipase down triglyceride into free fatty acids. These fatty acids can be transported across the intestinal membrane. However, once they cross the membrane, they are recombined to form again triglyceride molecule. Within the intestinal cells, these triglycerides are packed along with the cholesterol molecule in phospholipid vesicles called as chylomicrons. The triglyceride molecules represent the major form of the storage and transport fatty acid within the cells and in the plasma. The liver is the central organ for fatty acid metabolism. Fatty acids occur in liver by hepatocellular uptake from the plasma and by synthesis. To obtain the energy from the fat, triglyceride must first be broken down by the hydrolysis into their two principal component fatty acids and glycerol. This process is called as lipolysis. Takes place in the cytoplasm. The resulting fatty acids are oxidized by the beta oxidation into acetyl-CoA enzyme, which is used by the Krebs cycle. The chylomicrons enable the fat and the cholesterol to move within the aqueous environment of your lipotic and circulatory system. They leave the enterocytes by the exocytosis and enter the lipotic system via lecithin in villi of the intestine. Once in circulation, they can either go to the liver or be stored in fatty cells that comprise adipose tissues found throughout the body. This chylomicrons contains triglyceride, cholesterol molecules and apoproteins, protein molecules. They function to carry these water-insoluble molecules from intestine through the lymphatic system and into the bloodstream, which carries the lipids to adipose tissue for storage. Lipolysis. To obtain the energy from the fat, triglyceride must first broken down by the hydrolysis into two principal components, fatty acids and glycerol. This process is called as lipolysis. Takes place in cytoplasm. Resulting fatty acids are oxidized by the beta oxidation to acetyl coenzyme, which is used by Krebs cycle. The glycerol that is released from the triglyceride after lipolysis directly enters the glycolysis pathway as DHAP. Because one triglyceride molecule eats three fatty acid molecules within as such of 16 or more carbons in each one, fat molecules yield more energy than carbohydrates and an important source of energy for the human body. Triglyceride yield more than twice the energy per unit mass when they compare to the carbohydrates and protein. Therefore, these glucose levels are low. Triglyceride can be converted into acetyl coenzyme molecules used to generate ATP through aerobic respiration. The breakdown of fatty acids is called as fatty acid oxidation or beta oxidation begins in cytoplasm where fatty acids are converted into fatty acyl coenzyme molecules. This fatty acyl coenzyme A combines with a carnitine to create the fatty acyl creatinine molecule, which helps to transport the fatty acid across the mitochondrial membrane. Once inside the mitochondrial matrix, the fatty acyl carnitine molecule is converted back to the fatty acid, the acyl coenzyme, and into the acetyl coenzyme. Newly formed acetyl coenzyme enters the Krebs cycle and used to produce ATP in the same way as acetyl coenzyme derived from pyruvate. Ketogenesis. If the excess of acetyl coenzyme is created from the oxidation of the fatty acid and the Krebs cycle is overloaded and it cannot handle it, the acetyl coenzyme is diverted to create ketone bodies. These ketone bodies can serve as a fuel source if the glucose levels are too low in the body. Ketones serve as a fuel in times of prolonged starvation or when patients suffer from uncontrolled diabetics and cannot utilize most of the circulated glucose. In both cases, fat stores are liberated to generate energy through Krebs cycle and will generate the ketone bodies when too much acetyl coenzyme accumulates. In this ketone synthesis reaction, excess of acetyl coenzyme is converted into hydroxymethyl glutyl coenzyme A, which is a precursor for cholesterol and is an intermediate that converted into the beta hydroxybutyrate, the primary ketone body in the blood. Ketogenesis, excess of acetyl coenzyme is diverted from the Krebs cycle to the ketogenesis pathway. This reaction occurs in mitochondria of the liver of the cell. The result is the production of beta hydroxybutyrate, the primary ketone body found in the ketone body oxidation. Organs that have classically been thought to be dependent slowly on glucose, such as the brain, that can actually use ketone as an alternative source energy. This keeps the brain functioning when glucose is limited. When ketones are produced faster than they can be used and they can be broken down into carbon dioxide and acetone. The acetone is removed by exhalation. One symptom of ketogenesis is that the patient breath smells sweet like alcohol. This effect provides one way of telling if the diabetic is properly controlled in the disease. The carbon dioxide produced can acidify the blood, leading to the diabetic ketoacidosis, a dangerous condition in diabetics. Ketone oxidize to produce energy for the brain. Beta hydroxybutyrate is oxidized to acetoacetate and NADH is released. As HS coenzyme A molecule is added to acetoacetate, forming a acetyl coenzyme A. The carbon within the acetocoenzyme A that is not bonded to a coenzyme A then splitting the molecule into two. This carbon then attaches to the another free HS coenzyme A, resulting in two acetyl coenzyme A molecules. These two acetyl coenzyme molecules are then processed through the Krebs cycle to generate energy. Ketone oxidation. When a glucose is limited, ketone bodies can be oxidized to produce the acetyl coenzyme can be used in the Krebs cycle to generate respiration. The breakdown of fatty acids is called as fatty acid oxidation or beta oxidation begins in cytoplasm where fatty acids are converted into fatty acyl coenzyme molecules. This fatty acyl coenzyme A combines with a carnitine to creatinine to create the fatty acyl creatinine molecule which helps to transport the fatty acid across the mitochondrial membrane. Once inside the mitochondrial matrix, the fatty acyl carnitine molecule is converted back to the fatty acid acyl coenzyme and into the acetyl coenzyme. Newly formed acetyl coenzyme enters the Krebs cycle and used to produce ATP in the same way as acetyl coenzyme derived from lipogenesis. Lipogenesis begins with acetyl coenzyme, advances by the addition of the two carbon atom from another acetyl coenzyme. This process is repeated until fatty acids of appropriate length. Because of this is a bond creating anabolic processes, ATP is consumed. However, the creation of the triglyceride and lipids is an efficient way of storing the energy available in carbohydrate. Triglycerides and lipids, both high energy molecules, are stored in adipose tissue until they are needed. Although lipogenesis occurs in cytoplasm, the necessary acetyl coenzyme is created in the mitochondria and cannot be transported across the mitochondrial membrane. To solve this problem, pyruvate is converted into both acetoacetate or acetyl coenzyme A. Two different enzymes are required for this conversion. Oxaloacetate forms via the action of pyruvate carboxylase, whereas the action of pyruvate dehydrogenase creates acetyl coenzyme A. Oxaloacetate or acetyl coenzyme A combines to form citrate, which can cross the mitochondrial membrane and enters the cytoplasm. In cytoplasm, citrate is converted back to the oxaloacetate and acetyl coenzyme. Oxaloacetate is converted into malate, then into beta. Pyruvate crosses back across the mitochondrial membrane to wait for the next cycle of lipogenesis. The acetyl coenzyme is converted into mallyl coenzyme that is used to synthesize fatty acids. Lipids are available to the body from three sources. They can be indigested in the diet, stored in adipose tissue of the body, or synthesized in the liver. Fats indigested in the diet are digested in small intestine. Triglycerides are broken down into monosaccharides and free fatty acids, then imported across the intestinal mollusca. Once across the triglyceride are resynthesized and transported to liver or adipose tissue, fatty acids are oxidized through fatty acid or beta oxidation into two carbon acetyl coenzyme molecules which can then enter the Krebs cycle to generate ATP. If excess of acetyl coenzyme is created and overloads the capacity of the Krebs cycle, the acetyl coenzyme A can be used to synthesize ketone bodies. When a glucose is limited, ketone bodies can oxidize and used for the fuel. The excess of acetyl coenzyme A generated from excess glucose or carbohydrate ingestion can be used for fatty acid synthesis or lipogenesis. Acetyl coenzyme A is used to create lipids, triglycerides, steroid hormones, cholesterol, and bile salt. Lipolysis is the breaking down of triglyceride into glycerol and fatty acid, making them easier for body to process. Thank you. All the best.